Hello, my name is Mariana Nery. I'm very glad to be here today talking a little bit about using genomes from dolphins and manatees to track convergent evolution of fluvial colonization. That is one of the research lines conducted in my laboratory here at University of Campinas, Brazil. And here we are in this great session called Challenges, Opportunities and Advances in Latin American Genomics. And I want to thank the biodiversity genomic organizers for putting this session together, and of course for inviting me. It's a great honor. But I'm going to apologize to you because in my brief talk, I'm going to invert the order of the title of the session, starting by talking where we are here in Latin America, some advances we have made, some opportunities, and finally ending with some challenges. Let me start by telling you from where I'm talking. I speak directly from the city of Campinas, which is here in southeastern Brazil, the largest country in Latin America. In the city is located the University of Campinas, which is among the best in Latin America and has a very nice campus. You are all very welcome to visit us. I'm the PI at the Laboratory of Evolutionary Genomics, which is located in the Institute of Biology. Here are two pictures of our institute. So our group is interested in investigating the genomic basis of phenotypic biodiversity and the role of evolutionary forces in creating and shaping this diversity. We are interested in questions regarding the origin of groups, what mechanisms act on the origin of evolutionary innovations and adaptations, and also in convergent evolution. Our projects do not focus on a specific group, but we work mainly in aquatic vertebrates, such as penguins, sea turtles, and mainly in aquatic mammals. One of our main projects now is about convergent evolution, which is essentially when two species independently evolve to be similar. And it's a phenomenon known for a very long time across the whole tree of life. But it was until recently, with the massive amount of molecular data that has come forward in the last 2 decades or so, that revealed that convergent evolution at the molecular level is a more common phenomenon that we saw before. Aquatic mammals are a textbook example of convergent evolution. Independently and in different periods, terrestrial ancestors from cetaceans, they are dolphins and whales, sirenians, they are the manatees, and pinnipeds, sea lions, walrus, and seals, they start to explore new aquatic environments, and turns out, millions of years later, they were the magnificent animals that we love today. These animals, despite being very cute and awesome, they also share interesting similarities on their evolutionary histories, making the transition from land to water, evolving several similar characteristics such as streamlined bodies, they have lost legs, they have flippers, and so on, what make them natural evolutionary replicates. Convergent evolution, these particularly 3 groups, was explored before by Foot and collaborators, and also by Zoe and collaborators a few years ago, but many specific questions remain to be answered, especially between sirenians, the manatees, and cetaceans, which are the aquatic mammals that live exclusively in water, and because of that, they share even more similarities. And notably, both groups secondarily occupied fresh water. So, all these examples bring the ultimate question. After all, how creative is evolution? And how this convergence arises in a molecular level? What's happening in the genomes of these groups on all these habitat transitions? In Central and South America, we have 2 sister groups of cetaceans and sirenians that we chose to answer some specific questions. The 2 dolphins are the coastal Sotalia guianensis and the riverine Sotalia fluviatilis, and the 2 manatees are the coastal Trichechus manatus, and the riverine Trichechus inunguis. These two groups split and diverged by the same time, back from 2 to 4 millions of years ago during the Pleistocene. And interestingly, we have hybridization cases reported for Trichechus, and to my knowledge, not for Sotalia yet. So we are working with two main research lines in my laboratory, basically seeking these two main questions. What are the evolutionary and genetic mechanisms behind the origin of new species and groups, specifically during the split from the ancestor of the two Sotalia and from the ancestor of the two Trichechus? And also, what is the role of molecular convergence on phenotypic evolution, but now looking for the marine species and the fluvial species here showed. In which level this molecular convergence occurs? The same amino acids, the same sites, same genes, different genes, but same function. To answer that, we use comparative genomics, and so the first step is to sequence all these genomes. Sotalia guianensis, fluviatilis, Trichechus manatus, and Trichechus inunguis. We started this project 6 years ago when it was all about short reads, and by then we used pair-end and mate-pair strategies in Illumina platform to generate many reads with great coverage for each genome. But still, of course, we have 4 genomes very fragmented. With the new sequencing technologies, we want to improve the fragmentation of the Sotalia genomes, both the Sotalia genomes. For high-C sequencing, we needed a cell culture, and the great collaboration with my dear friend Susana Caballero from the Universidad de los Andes, who was able to culture the cells of Sotalia dolphin from an aquarium in Colombia, allowed us to be generating high-C data right now. In collaboration with other groups here in Brazil who provided skin samples, also allowed us to generate PacBio data. With this, we hope to achieve a reference genome at the chromosomal level for Sotalia very soon. Regarding the Trichechus species, what was available in public databases until recently was not a high-quality genome. For Trichechus manatus, we have greatly improved the coverage by resequencing the genome, and for Trichechus inunguis, the Amazonian one, we have generated this data for the first time. And although the genome that we generate, both genomes that we generated, are still fragmented, it is still much better than we had before. A little less than a month ago, at the end of August, the DNA Zoo project released the chromosome level genome of Trichechus manatus. So now my students are mapping our contigs onto the now reference genome of Trichechus. The expectation is that we will greatly improve the fragmentation of our genomes to proceed with the analysis. But even with a fragmented genome, we were able to extract important information. One of them was to recover the complete mitogenome of the 2 Trichechus species we studied, and to investigate the evolutionary history of the whole Trichechus genus group. We also sequenced the mitogenome of the African manatee and published this paper last year, The Evolutionary History of Manatees Told by Their Mitogenomes. And with the whole genomes of the coastal manatee and the Amazonian manatee, we made all the basics on genome characterization and genome analysis. I will not go into to get into much details here today. Just to let you know, despite the level of fragmentation, we were able to produce good results, such as the transposable elements landscape, an upset plot to show the overlap and unique set of genes belonging to each Trichechus species, and to explore the protein coding genes unique to the Trichechus genus, and those shared with at least one other Afrotheria. We also performed a functional annotation to infer overrepresentation in the Trichechus exclusive group compared to other Afrotheria. We also estimated the divergence time of each species using single copula or talux, performed analysis of gene families, expansions and contractions, and inferred historical population sizes using PSMC. But as I don't have much time here today, and as we will redo all this analysis after having improved the fragmentation of our genomes using the Zoo DNA the DNSU reference genome. I will not show you the details of the results today, but maybe next year at the same Congress. Talking about opportunities, I personally think that these are exciting times for evolutionary biologists, as genome scale data sets are applied to a never expanding range of problems and also to fundamental questions regarding the origin and evolution of biodiversity. So I think that there are plenty of opportunities and many data available to answer interesting questions about the evolution of so many species, including neotropical species. And of course, the costs are not prohibitive as they usually were. In our laboratory specifically, we are taking advantage of many cetacean and other mammalian genomes available to study genes in regulatory regions related to echolocation, immunity, sense, metabolism, body size, cancer, osmoregulation, and hypoxia. And also, we are going beyond the genes, working also with microRNAs and transposable elements. About the challenge, well, I could talk about several challenges in Latin America in the field of genomics. Some samples are difficult to obtain in the quality that we need for a reference genome. In our case, we work with aquatic mammals and we mostly depend on animals in captivity, but this is not unique to our region. I could also talk about the terrible decrease in investment in science, at least here in Brazil. Even though the cost of sequencing have dropped a lot, it is still not possible in many regions of our country to perform this sequencing because the laboratories depend on funding from the federal government only. Of course, that the expertise is also something important. The formation of human resources qualified to work with comparative genomics is very important in our region through courses that can train our graduate students in this analysis. But I would like to highlight a very big challenge, particularly for our project, which is bureaucracy. The project that I presented to you started many years ago, and progress has been slow and is still happening a lot because of the bureaucracy involving sending samples to companies outside of our countries to perform the sequencing. When we have quality samples, which is what we had from Colombia, when you have the money, when you have the expertise, we had this big obstacle that were the papers and all the documentation to send the sample to the United States from Colombia, which took 1 year and 3 months to happen. So that's where are right now, waiting for the data that are being produced right now. Anyway, the new tropics is one of the areas of greatest biodiversity on the planet, and that's not new for any of you, and also a great concern regarding the conservation of the species. Of course, even though we do not live in the best possible scenario in terms of investment and other problems that I mentioned, I think that no country or region will ever be in the best possible situation. And this does not discourage us from continuing because resilience and insistence are important characteristics for the Latin researcher. Well, this is what I had to tell you today. Thanks again for the organizers for this amazing Congress. Thank you all to all my students and collaborators, and thank you to all of you. Here's my email in case you want to get in touch. Bye-bye.